Hello, everyone. Welcome to this podcast. In this podcast, we're going to talk about the first part of Chapter 7. This is was covered on day 9. Remember, during these podcasts, at least most of them, and particularly this one and the next couple, really what we want you to do is take out a blank sheet of paper and draw and write things that we write on the board here. And in this way, we're going to cover some of the main topics from the chapter, including the main topics from the note-taking guides. This isn't meant to substitute for the note-taking guide. The note-taking guide is still your primary resource for studying the material and preparing for the exams. But what we've discovered is that this textbook, the chapters are pretty dense, and sometimes it's hard to pull out exactly what it is that we want you to have, particularly how deep to go into any given topic. So this should help out. Help you all out in deciphering what we believe is important in these chapters and how deep to go. In this first part of this chapter, of chapter 7, we talked about phospholipids. In the first part of this chapter, we talked about phospholipids. And we often will draw a phospholipid showing the phosphate hydrophilic head with a circle, and then the two hydrophobic fatty acid tails like so. So let's go ahead and label these. And throughout this, I'll often use some variations of shorthands, and I'll try to explain to them when I do it. But for hydrophilic, I'm just putting an H dash and then the word philic. This will help save time as we're writing these. So that's for this phosphate hydrophilic head. And then over here, we have the hydrophobic fatty acid tails. And for this, I'm going to write an H and then a dash phobic. Now, when you get a whole bunch of these phospholipids together, what they will form is a phospholipid bilayer. Okay, so I'm just going to draw a few of these. And we said it's a bilayer, so bi for 2. And so we're going to have the other phospholipid groups right here. And the reason they formed this bilayer is because the hydrophobic portions of the fatty acid tails like to interact with each other. And so they naturally come together, forcing the hydrophilic portions on the outside of the membrane. Now, an important part of this is that now that we have the hydrophilic portion on the outside and the hydrophobic inside, and remember we have water out here, this makes this membrane itself soluble in the water, and that's important. And it's soluble because it's interacting with the water now. As we look at this membrane, we know that, again, this outer portion, hydrophilic, and this the phosphate is hydrophilic here as well. And this region, the middle of the membrane, is hydrophobic. And I'm just going to write phobic here. Is these hydrophobic regions that allow this membrane to form. It's probably the most important part of it. Now I'm going to draw another membrane over here real quick. When we think of the phospholipid bilayer, we think of it as having two components. The phospholipids themselves and the membrane also have proteins associated with them. And these proteins can be one of two broad classes. They can be peripheral proteins, and we'll draw that in, how about blue? I'm just going to write the initials P and then another P. But in your notes, you might want to go ahead and write out peripheral protein. These proteins interact with the phosphate groups of the phospholipids, and they can be on either side. The other class of proteins associated with membranes are called integral proteins. And let's draw those in, how about green? And I'm just going to write IP here for integral protein. These can be found in a few different locations. They can be found right here in the inside of the membrane in the hydrophobic regions. And let's go ahead and draw a few more phospholipids here to show you where the other integral membrane proteins could be. I'm going to leave some gaps here. You'll see why in a moment. Okay, now that we have the additional phospholipids drawn here, let's go ahead and draw two other types of integral proteins. This one we're going to call a transmembrane protein. Transmembrane because it goes all the way across the membrane, like so. It is an integral protein because it goes inside the membrane, but we also can call this a transmembrane protein. So I'm going to just write T period M for transmembrane, and then dash P for protein. It's a class of integral membrane protein, but since it goes all the way through the membrane, we call it a transmembrane protein. Now, some integral membrane proteins, what you'll see is they'll be on one end of the membrane, and they'll just be partway into the membrane like shown here. This is also an integral membrane protein. Anytime that protein partially or completely goes into the membrane, we call it an integral membrane protein. One other thing your book talks about is the fact that these proteins often have sugars associated with them. And they can have elaborate sugar components to them. For simplicity, I'm just going to draw a single G for a single glucose. But you should recognize that many different carbohydrates can be attached to these proteins. Either the peripheral protein or the integral membrane proteins. Every now and then, these can be found attached to the lipids. When they're attached to the proteins, we call them glycoproteins. When they're attached to the lipids, we call them glycolipids. These are important in the cells, our cells, being able to self-recognize our own cells so that we can tell the difference between our safe cells that are part of our body versus cells that are foreign invaders. And then if they are the foreign invaders, then we have our immune system which will go after them and kill those foreign invaders. So these are the most important parts of the membrane. Now, we didn't mention it, but we also have cholesterol that can be found inside of the membrane as well. And we'll draw that here as a magenta. And I'm just going to draw it as a ring structure, like so, and put a C in it. And this cholesterol will be floating in the membranes, and it can affect the fluidity of the membrane and has other important functions as well. While we're talking about fluidity, though, let's go ahead and mention a couple features about fluidity that your book mentions. Remember, as we discussed in class and as your book highlights, the membrane itself is not a static structure. It's very fluid. It changes all the time. It can change depending upon the environment outside. It can change depending upon the organism you're talking about. And because of that, the fluidity changes. As I said, it changes. But how does it change? Here are a couple key things to remember. I'm going to draw an up arrow. Up arrow for me is just showing an increase. So increase in fluidity. And for simplicity, I'm just going to write fluid. So increase in fluidity. You get this when you have an increase in unsaturated fatty acids. And for simplicity, I'm just going to write it like so. So again, remember that the unsaturated fatty acids, if we were to draw them in a quick diagram, you'd have your phosphate group here. Unsaturated means that some of those carbons don't have hydrogens all the way around, and so it forms these kind of kinks in the lipid. So we'll signify that by doing this. So here's a good way to diagram a fatty acid that has unsaturated carbons on it. Remember, these are carbons that don't have hydrogen all around them. Just like we see in unsaturated fatty acids like vegetable oil, which is a liquid, remember it's a liquid because there's these kinks in the carbon-hydrogen chain. These kinks prevent it from stacking nicely and neatly and prevent it from being a solid. And so just like in our membranes, more unsaturated fatty acids will allow the membranes to be more fluid. Now if we have a decrease in fluidity, I'm just going to abbreviate it by fluid, we will have more saturated fatty acids. And we could signify that by just showing a single phosphate with the two fatty acids and drawing them as straight lines. Remember, these carbons are fully saturated with hydrogens, so they form these very straight and rigid carbohydrate chains, and they stack really nicely and neatly on them, just like you would see with animal fat, a solid structure. When this occurs in the membrane, it becomes less fluid, that is, more solid. Your book also discusses what cholesterol does in the membrane and how it influences fluidity. It has different effects at lower or higher temperatures, and I'll let you look in the book to see if you can figure out what it does. In essence, what it does is it will interact with the fatty acid chains, and it will help kind of clog them up and restricts their movement, and that in the end will also influence fluidity. The last part of the study guide, the note-taking guide, talks about transport proteins and diffusion across this membrane. So I'm going to extend our membrane out a little bit more. Now, nonpolar molecules like oxygen or CO2, these are nonpolar molecules. Both of these will readily dissolve into the membrane and pass through and exit the other side. Again, these are nonpolar molecules, and they have no problems passing through a membrane all by themselves. Now, where we have problems is when we have, I shouldn't say problems, but where it becomes less easy is when the molecules trying to get across the membranes are ions, something say like calcium, or like water. So these charged or polar molecules cannot just readily diffuse across the membrane. Rather, they require specific transport proteins. And now we can come back to our good friend here, the transmembrane protein. And sometimes these transmembrane proteins can serve as these transport proteins. And in this case, let's just say that's what it is. And now this calcium and the hydrogen, for instance, could go across this transport protein to the other side. These transport proteins are often very specific and will only allow certain classes of molecules to go across. But again, they would be your polar molecules or small charged ions, like we see here. All right, that is the main part from the first part of Chapter 7. If you have any questions as you're going through the note-taking guide or the material in the textbook, please make sure you come see one of the instructors, and we'll be happy to help you out. Good luck.